Imagine having molecular scissors so incredibly precise that can find and snip out a single word in a massive book. A word buried deep within countless pages. That's essentially what CRISPR does, but instead of words, it targets DNA, the very blueprint of life. CRISPR, remarkably discovered in bacteria, lets scientists edit genes with unprecedented precision, opening up possibilities we once only dreamed of. Bacteria use CRISPR as a defense, a shield against invaders, storing bits of viral DNA like digital fingerprints, allowing them to recognize and neutralize future attacks with remarkable efficiency. Scientists, with their insatiable curiosity, realized this natural system could be repurposed to edit any DNA, plant, animal, or human. The key to this incredible precision? A guide molecule that acts like a GPS, finding the exact target sequence, and a protein called Cas9, which acts as the molecular scissors, doing the cutting with pinpoint accuracy. By carefully designing the guide molecule, we can direct Cas9 to any gene we want to change, offering unprecedented control over the genetic code. This groundbreaking breakthrough, spearheaded by the brilliant minds of Emmanuelle Charpentier and Jennifer Doudna, rightfully won them a Nobel Prize, and has irrevocably revolutionized the field of genetics. CRISPR isn't just a lab invention, a product of human ingenuity. It's a natural wonder, a testament to the power of evolution that we've learned to harness for the betterment of science and potentially, humanity. Now, armed with this knowledge, we stand at the precipice of a new era, where we can rewrite the very code of life itself. So how does CRISPR actually work its magic? What are the key components that make this revolutionary gene editing possible? Let's break down the process and explore the fascinating mechanisms behind CRISPR technology. It's a two-part system, a dynamic duo working in perfect harmony to achieve precise gene editing. These two crucial components are a guide RNA and the Cas9 protein. Think of them as a smart bomb precisely guided to its target. The guide RNA acts like a GPS, providing the precise coordinates and leading Cas9 directly to a specific DNA sequence within the vast genetic landscape. It ensures that Cas9 goes to the exact location that needs editing. Once it arrives at the designated spot, Cas9 then acts like molecular scissors and cuts the DNA at that exact spot, creating a break in the double helix. The cell's natural repair system, always on standby, immediately jumps in to fix the break, sometimes disabling a gene completely, effectively turning it off. But here's where it gets even more interesting. Scientists can also provide a new DNA template, which the cell uses to fix or replace the original sequence, essentially rewriting the genetic code. Think of it like correcting a typo in a document. Find quick, cut it out, and paste in the correct spelling, quick. This powerful ability lets us fix genetic mistakes that cause devastating diseases like sickle cell anemia, offering hope for potential cures. But it's not just about fixing existing problems. CRISPR can also insert entirely new genes, opening up possibilities for genetic enhancement. In agriculture, we can create crops that are more resilient, able to resist drought or pests, simply by adding helpful genes that improve their survival and yield. The process is remarkably precise, surprisingly fast, and fundamentally based on nature's own ingenious toolkit. CRISPR gives us unprecedented control over life's very blueprint, offering both immense potential and significant responsibility. CRISPR's impact is already being felt across numerous sectors. Its potential to reshape our world is rapidly becoming a reality, most notably in medicine and agriculture, where it's showing immense promise. Clinical trials are actively using CRISPR to develop innovative treatments for a range of genetic diseases. These trials represent a significant step forward in gene therapy. Consider sickle cell anemia, for example. CRISPR offers a groundbreaking approach by correcting faulty genes directly within patients' own cells, offering the potential for a lasting cure. It's also being rigorously tested to supercharge immune cells, enhancing their ability to recognize and destroy threats. This is particularly relevant in the fight against cancer, offering new avenues for immunotherapy. Researchers are exploring its use to remove HIV from infected cells, potentially leading to a functional cure for the virus. In agriculture, CRISPR is revolutionizing crop development, helping to create crops that are not only more nutritious but also more resilient to environmental stresses, and ultimately more sustainable for our planet. Take fungus-resistant wheat, for instance. Or vitamin-rich rice, designed to combat nutrient deficiencies in vulnerable populations. Scientists are even employing gene editing techniques on mosquitoes. The goal is to block the transmission of devastating diseases like malaria, which disproportionately affects communities around the globe. These advances hold the potential to significantly improve global food security and strengthen our defenses against future pandemics. CRISPR is fundamentally transforming how we approach and tackle some of humanity's biggest and most pressing challenges, offering innovative solutions where few existed before. The possibilities are vast, constantly expanding, and promise a future shaped by scientific innovation. With CRISPR's power comes big responsibility. Editing human embryos could erase genetic diseases, but also raises ethical questions about designer babies and inequality. Mistakes or unintended effects could be passed down for generations. The same concerns apply to editing plants and animals. What if changes spread in the wild? That's why careful regulation and public discussion are crucial. CRISPR can solve huge problems, but we must weigh risks and benefits. It's not just a scientific issue, it's about the future we want to create. Everyone should understand CRISPR and help guide its use. The future is in our hands. Let's use these scissors wisely. Like, subscribe, share, and support to the greatness. 19 channel and click in the description box below for products and services.